Hello, hello! This video segment is dealing with your Chapter 20. So we're going to do a little bit of review, and I'll just highlight a few things coming from your PowerPoints. This chapter included some groups, Vibrio, the Aeromonas, Plesiomonas, and Campylobacter species, and then one particular species, which was Helicobacter pylori. So let's just jump in. Let's start with Vibrio. Vibrio species are found worldwide in aquatic environments. They can cause pandemics, and these have been reported since 1817. There has been an increase in isolation in the laboratory of Vibrio species over the last several years, and this is due to increase in travel, increase in consumption of seafood, especially raw seafood, increase in recreational water facilities, and there's a heightened awareness in the laboratory about these. So we're always on the lookout for Vibrio species. So there are some clinical manifestations to the disease that Vibrio causes, which is cholera. And some of these manifestations include consumption of raw seafood, immigration or travel to foreign areas that have an epidemic or have been known to have epidemics of cholera, gastroenteritis with cholera-like or something called rice water stools. These rice water stools have flecks of mucus, so this is very telltale for this particular infection, these rice water stools. Accidental trauma during contact with fresh or marine water. So for example, you step on a shell, or you cut your foot on a rock while in the water at the sea. Cholera tends to increase in summer months, of course, because people are outside and playing in water. It makes sense. And then, of course, there's a decrease in the winter months. So, some morphology characteristics of Vibrio species. They're gram-negative, they're non-spore forming, they're little tiny rods, little tiny gram-negative rods. When grown in broth, they have a polar flagella. When grown on solid media, they will develop peritrichous flagella, which means the flagella surround the entire organism. These little tiny Vibrio have been described as curved rods seen on the initial gram stain. However, they tend to look more straight when cultured rather than curved like they are in its natural environment. So after growing them in the lab, they tend to lose their natural curve. So all Vibrio are oxidase and nitrate positive. They are sensitive to something called O129, O/129. This is a compound called 2,4-diamino-6,7 disopropylpteridine. That's a mouthful, but anyway, that's what O129 stands for, and Vibrio are sensitive to this, so they will be destroyed or killed with this particular agent. Another unique characteristic or quality about Vibrio physiology is that they are positive to something called the string test. So when the organism is mixed with a 0.5% sodium desoxycholate, it forms an agglutination. And when you pull that little string up or rod out of your suspension of organism and desoxycholate, it will form a string. It presents as a string of agglutination. So know that it's positive for the string test. And important important enough, all species except for Vibrio cholerae and Vibrio mimicus are halophilic, so they are salt-loving. So let's talk about antigenic structure and some of the epidemiology and virulence that goes along with Vibrio and cholera. All strains of Vibrio cholerae share a common flagellar antigen, which is the H antigen. So all Vibrio cholerae share the same H antigen. However, cholera vibrios are further divided into 6 serogroups based on their somatic O antigen. And of these 6 strains, or serogroups, pardon, strains of Vibrio cholerae that agglutinate with the somatic antigen O1 antisera are associated with epidemic cholera. So strains that resemble Vibrio cholerae but fail to agglutinate with the O1 antisera are called non-O1 Vibrio cholerae strains, and these are not epidemic. So the Vibrio cholera are determined into 6 sub-serogroups, and of those 6 groups, the ones that contain the somatic antigen O1 are virulent and cause epidemics. And of those O1 strains, they can be further divided into 2 biogroups, classic and LTOR. So just know that there's 2 biogroups of the O1 strains of Vibrio cholerae, and these are what cause your epidemic cholera. Let's see. There are 4 major Vibrio species you'll encounter in the laboratory. Of course, that first one I mentioned, Vibrio cholerae. You have your O1 and your non-O1 strains. You'll see a species called Vibrio parahemolyticus. You'll see a species called Vibrio vulnificus and Vibrio alginolyticus. So we're going to talk about all 4 of these species. The first one we're going to talk about is Vibrio cholerae, most serious Vibrio associated with infection. Know that. This one's the most serious species that goes along with epidemiology. So this particular species, Vibrio cholerae, is also referred to as Asiatic or epidemic cholera. Most cases occur in developing countries, and it's rarely reported here in the U.S. It's spread mainly through contaminated water. With this infection, you'll have those rice water stools with numerous mucus flecks. You'll have 10 to 30 stools per day. You can lose up to 20 liters of fluid per day. So with rapid fluid loss, you have rapid electrolyte loss. This becomes a very serious situation, can cause death. So this is one of the main concerns with cholera, is dehydration. Also, Vibrio cholerae can produce a very powerful enterotoxin known as the cholera toxin, or a choleragen. So when you're infected, or they determine that you've been infected with Vibrio cholerae, you have an aggressive fluid replacement treatment with antibiotics, and hopefully that patient will recover. The second species of Vibrio is Vibrio parahemolyticus. This is the second most commonly isolated Vibrio causing gastroenteritis. It's not the second most serious, it's just the second most commonly isolated. The next species we talk about, which will be Vibrio vulnificus, that is the second most serious that's associated with infection. But parahemolyticus is the second most commonly isolated, second to Vibrio cholerae. It's the #1 cause of summer diarrhea in Japan. It's found in aquatic areas and has been isolated in nearly every coastal U.S. state. Most gastroenteritis cases are traced to the consumption of raw, improperly cooked, or contaminated seafood, especially oysters. So if you like oysters, be careful. If you ever develop signs and symptoms of gastroenteritis after eating them, maybe this might be something they should screen your stool for. So it is usually a self-limiting infection. You'll have watery diarrhea, moderate cramps, or vomiting. There is this thing called a Kanawagha phenomenon, and this is hemolysis produced by Vibrio perihemolyticus that indicates a more virulent strain of this particular organism. So just be familiar with that little name, Kanawagha phenomenon. So let's move to the next species. This is our third one, and this is the second most serious associated with infection. And this is Vibrio vulnificus. Let's see. Infections generally fall into two categories: primary septicemia and wound infections. So the primary septicemia is usually of the GI tract route caused by shellfish. There is a 40 to 60% mortality rate, so this is pretty serious infection. There's also the wound infection that happens to come along with possibly a traumatic wound infection near aquatic areas. The fourth species is Vibrio alginolyticus. This is least pathogenic to humans, least commonly isolated. It is a strict halophile. Nearly all infections originate from extraintestinal sites, such as the eye, the ear, wound and burn infections. The organism may also be an occupational hazard for people that work near seawater. So in order to collect and transport Vibrio, there's no special growth requirements other than the attention to salt content in the media. Most pathogenic Vibrios are lactose non-fermenters, kind of like our true Enterobacteriaceae pathogens. They're non-lactose fermenters. It's very important to check oxidase reactions of suspicious colonies. And remember, Vibrio are oxidase positive. There is a particular agar called thiosulfate citrate bile salt sucrose agar, or TCBS. And this differentiates our different Vibrio species. So it's a very selective medium used to isolate Vibrio from stool. Your cholerae and alginolyticus, sorry, that is a hard word for me to say, ferment sucrose and produce yellow colonies, whereas perihemolyticus and vulnificus do not ferment sucrose and produce blue-green colonies. So, just to sum up your Vibrio, they're oxidase positive, they reduce nitrates to nitrate, so they're nitrate positive, they're sensitive to O129 compound, and they're unable to ferment inositol. So that's Vibrio. Let's move to our next group, and this is Aeromonas. The Aeromonas are also oxidase positive. They're widely distributed in freshwater, estuarine, and marine environments. Estuarine pertains to an estuary, which is a partially enclosed coastal body of water where one or more rivers or streams flow into this brackish water, and then it has a free connection to the sea. So it's kind of a mixture of salt and fresh water. These Aeromonas species are glucose fermenting, and they are straight gram-negative rods. Most of them are motile by a single flagellum. Aeromonids are classified into two groups. The first one is a mesophilic group. So a mesophile is motile at 37 degrees Celsius, which is our body temperature. And then there's a second group called the psychrophilic group, which grows between 22 and 25 degrees. And these are non-motile. So just know that the mesophilic group is motile at body temperature, and then the psychrophilic group is non-motile, growing at a little bit colder temperatures, room temperature. But all aeromonids typically grow between 4 and 42 degrees Celsius. They are an intestinal infection causer. Not always, but usually involves water exposure. There are 5 diarrheal presentations observed with this intestinal infection. There's an acute secretory diarrhea that has vomiting along with that. There's an acute dysenteric diarrhea with blood and mucus. There's a chronic diarrhea. There is a cholera-like type inducing rice water stools. And there's traveler's diarrhea. There's also wound infections that can be caused by Aeromonas. Usually a traumatic aquatic exposure is somehow involved. And the most common presentation of this form of infection from an Aeromonas is cellulitis. And the organism that's primarily responsible for most of these situations is Aeromonas hydrophila, that particular species. And an interesting note, there has been an association between Aeromonas infection and medicinal leech use. So just a fun fact there. So there is another infection that's fairly rare caused by Aeromonas, and that's septicemia. This is an invasive type of Aeromonas infection, and usually it is related to an infection with a species called Aeromonas vironii, Biovar sobria. So this is a particular biovariant of this species that can cause septicemia. And usually it's seen in people that are immunocompromised. Culture media. Usually Aeromonas grow on routine culture media, but it has a unique odor in that it smells like ammonia. So this is a unique quality of Aeromonas. Sometimes it's overlooked on the MacConkey because some species ferment lactose. So it's expected to be a non-lactose fermenter, but sometimes it does ferment lactose and could possibly be overlooked on that MacConkey. Aeromonas species are resistant to O129. So unlike Vibrio, which are susceptible, Aeromonas species are resistant to that O129 compound. So this is a way to differentiate or distinguish from Vibrio. They're oxidase positive, remember that, and this can distinguish them from Enterobacteriaceae. So I think that wraps up Aeromonas, except for the fact that they're usually resistant to penicillin and ampicillin, carbapenicillin, and cephalothin, or carbapenicillin and cephalothin. So we usually treat these infections with third-generation cephalosporin. So let's talk about the next group in this chapter, and this is the Plesiomonas species. Generally speaking, they're also oxidase positive, glucose fermenting, facultatively anaerobic, straight gram-negative rods. They're motile bipolar flagella, and there's one particular species called Plesiomonas shigelloides that causes human infection. Plesiomonas are intolerant to increased sodium chloride, so this makes this group different than the other two that we just discussed. They're found in fresh and estuarine water, so they can tolerate a little sodium chloride, but not much. They've emerged as a possible cause of enteric disease in humans, and they're similar to E. coli on most ordinary enteric lab media, so they look similar to E. coli. They cause gastroenteritis. Plesiomonas causes gastroenteritis. There's been at least 2 well-documented outbreaks in Japan. And this has been caused by ingestion of contaminated food or water. There are 3 clinical types of gastroenteritis that can be seen with infection with Plesiomonas. And this is the more common watery or secretory diarrhea. And there's a subacute or chronic disease that can last up to 14 days or even 3 months. And then there's a more invasive dysenteric form resembling colitis. So this is the single most common clinical symptom is abdominal pain. So you may have this and not even really know it. It's self-limiting. It can cause an extraintestinal infection that's related to occupational exposure. So it's not, that's not really a very common infection method, but or route, but it can be related to occupational exposure. Let's see. One other note, Plesiomonas ferments inositol, so this separates it from Aeromonas in nearly all Vibrio species. So this is a distinguishing characteristic. So let's continue on to our next group of organisms called Campylobacter. Campylobacter species have been known to cause abortion in domestic animals. Campylobacter jejuni, which is a particular species that is the most common cause of bacterial gastroenteritis worldwide. Know that. Infections have been attributed to animal exposure in handling infected pets. You can have indirect exposure by contaminated water and dairy products or improperly cooked poultry that can cause infection. And it has been seen in person-to-person transmission as well. Patients infected with Campylobacter jejuni usually begin with mild abdominal pain within 2 to 10 days after organism ingestion. They'll develop cramps, and then they'll develop bloody diarrhea. Illness is usually self-limiting, resolving in 2 to 6 days. Untreated patients may remain carriers for several months. Let's see. There is a particular agar called the Campy blood agar that we use to culture this organism. It is a Brucella-based agar that contains the acronym TVPAC, if you remember, antibiotics, which are trimethoprim, vancomycin, polymyxin B, and amphotericin B, and cefalothin. There's also another media called Skiromedium and Butzler medium, or medium V, that can also be used to culture Campylobacter species. They're quite fastidious and picky organisms, so incubation is a lot different for these organisms than with most other organisms we attempt to culture in the lab. We incubate these at 42 degrees Celsius, so slightly warmer than normal or body temperature. These organisms require microaerophilic, and they are definitely capnophilic. So we use a special gas and container or bag that we fill the gas into to create this ideal atmosphere, which it's 5 to 10% oxygen and 10% CO2, and the rest is nitrogen. We incubate these for 48 hours before we ever look at them. And usually the state monitors cases of Campylobacter. However, now there are some immunoassay enzyme tests that take 15 to 20 minutes. So it's a much faster method for detecting Campylobacter. If you look at Campylobacter species underneath the microscope, you will see a classic gull-wing appearance. These are curved gram-negative rods with this characteristic gull-wing appearance. They stain poorly with a gram stain, so often you stain them with carbofusion 2 to 3 minutes versus your normal, which is usually in half of that. They do express darting motility in saline under the microscope, so this is also a characteristic of Campylobacter. So gull wings and darting motility, important features. They're usually non-hemolytic and tan, slightly pink colored colonies. Let's see. The drug of choice to treat a Campylobacter infection is erythromycin. But most patients recover without any treatment. It is self-limiting most of the time. So that's that for Campylobacter. The last organism I want to talk about in this chapter is Helicobacter pylori. This particular organism is strongly associated with gastric and duodenal ulcers. H. pylori is recognized as the major cause of chronic superficial gastritis or type B gastritis. It's identified in as many as 75% of gastric ulcer patients and has been reported to colonize 20 to 40% of the U.S. population. Once acquired, this organism is able to colonize the stomach for a long period of time because it develops something called a biofilm. So it protects itself in this little tiny colony sac, so to say. It can cause a low-grade inflammatory process. And like I said, it's a major cause of group type B gastritis. And there is a very strong association between long-term H. pylori and infection, long-term H. pylori infection and gastric carcinoma. So it's important to detect this in patients. There were 2 individuals in Australia named Barry Marshall and Robin Warren, and this is just a fun fact, that won a Nobel Peace Prize for testing on themselves, is what it essentially was. They formed an ulcer on, one of them formed an ulcer on himself with this particular organism to prove that this organism was the causative agent of ulcer. And then, of course, they treated themselves and cured it. But yes, that was, those 2 individuals had made a big difference and impact in treatment for people all over the world. It can also be recovered, this organism, from gastric biopsies. They have to be transported quickly, but we use something called a Christensen's urea agar that if you have this urease producing organism on it, it will turn bright pink and become, that's what's known as positive. So it can be recovered on chocolate and scleromedium as well. It's very similar looking to Campylobacter under the microscope, actually. And it's very treatable with amoxicillin and, well, usually it's a triple therapy with amoxicillin, metronidazole, and tetracycline, I do believe. So we also, most commonly in the laboratory, and you will see these, is the rapid urease testing, which they call the CLO test. So they take a little tiny biopsy of your stomach area that they believe may be infected with H. pylori, and put it in this little tiny cartridge with agar, with Christensen urea agar. And so they just kind of close it up, and it's a real quick method, surest method for detecting Helicobacter pylori in a patient biopsy. There are some other ways to test for it. There's a urea breath test, and there is a Helicobacter pylori serum antibody test as well. So that is that chapter, chapter 20. So just kind of highlight those things in your notes and study that chapter. Know your most important aspects of each of those organisms. And that's a wrap for chapter 20.